All right. Hello and a warm welcome to everyone. My name is Oliver Mandal and I'm Senior Product Manager for Process Probes and Flow Cells at Hellma. I'm very happy to host today's webinar featuring our guest speaker and PAT expert, Joey Sloop. Together, we will explore the topic Unlocking the Power of PAT, 20 years insight into safer, smarter, and more efficient processes with NIR. Let me introduce Joey to you. With over 30 years in the chemical industry, Joey has extensive experience with process analytical technology. Throughout his career, he has worked across analytical, chemistry, process optimization, material analysis, and regulatory compliance. At SI Group, Joey wears a couple of hats as senior lab technician and PAT manager. For more than 20 years, he has dedicated his work to NIR and Raman spectroscopy, leading projects from system integration to data interpretation. One of Joey's recent achievements was the modernization of a 50-year-old production line through the integration of a new NIR system, enabling real-time monitoring, unlocking the potential annual yield increase of 250,000 pounds. And I'm glad that he takes time to share his experiences with us today. So welcome, Joey.
All right, guys. Good afternoon. Sorry for the technical glitches, but we got it going. And thank you all for attending. And thank you, Oliver, for the introduction and getting this thing rolling. I'd like to tell you briefly about how I got into process analytical technology, often referred to as PAT or PAT for short. For the last 33 years, I've worked in the chemical plant that makes antioxidants, fuel additives, flame retardants, catalysts, and pharmaceuticals. Most of that time has been spent in the quality control lab, but I've also served as material analyst and worked in environmental and regulatory control. At one point, I was asked to assist an R&D chemist. He had successfully implemented the installation of our first NIR system and was in need of help with his various projects, including NIR model building. After a couple of years, that chemist took a job with another company and the whole thing fell into my lap. And that is how I really began to learn process analytical technology and now have the responsibility for all aspects of these systems. During my time working with Pat, we have expanded to 4 NIR systems and 2 Ramans. We have seen many benefits and improvements as a result. And it is to expound these benefits and improvements that I've been asked to speak to you today. Now, it'd be a mistake to assume that everyone fully understands and might not need a little further explanation on the basics. So I'm going to start with some basics and fundamentals of process analytical technology. Then I will discuss the advantages that we have seen at my plant in the areas of safety, efficiency, and knowledge. And then I will briefly cover the differences in the cost of a NIR system versus the traditional GC. And finally, we will end with some typical arguments against PAT systems and why they are completely illogical. So, what is process analytical technology? Broadly speaking, it's any of the devices installed on process equipment which relates to the chemical technician process conditions. Examples include thermocouples, pressure transducers, flow meters, and inline pH probes. However, for this discussion, we'll be focusing on those systems which relay complex analytical results, such as component concentration, more specifically, NIR. The basic setup of a PAT system is an instrument, housed in a remote and environmentally friendly location, a probe which is placed in the process, and the fiber optics connecting the two. The first picture here is a NIR instrument with a 6-channel multiplexer. This particular instrument has the ability to analyze 6 different streams. The blue wires you see coming from the instrument are the fiber optics, one sending a signal, the other receiving. The second picture here is of a typical probe, in this case a transmission probe. The back end is where the fiber optics connect. The flange bolts the probe onto the process equipment, and the body of the probe extends into the process and reaction material. The slit near the end of the probe has a window on both sides. The chemical being analyzed flows through the slit. Light is transmitted through one of the windows, through the chemical, and back into the window on the other side of the slit, and then returns to the instrument. It is the change of light that passes back through the fiber optic cable that the instrument analyzes. The third picture here shows a standard probe placement in a process. Here, the probe is placed directly above a pump in a circulation line. Now, to give you a better understanding of how these instruments work, let's consider sight. Very generally, light is emitted from a source, the sun or a light bulb. Light then travels from the source to an object. Once the light hits an object, it then changes and reflects off the object. That changed light is what your eye receives and is interpreted by your brain. From that changed light, you can see color, shape, size, and detail. We can immediately gather a vast amount of information from what we see. You see a person and can tell their sex, height, approximate age and weight, long hair, short hair, no hair. You can get an idea of someone's general state just by looking at them, maybe even be able to tell what emotions they're feeling. NIR works the same way. Light is emitted, travels to a chemical, changes, and then returns to be analyzed. Now, take a look at this chart. Just slightly left of center, you will see the visible light spectrum. This is the light that we see with our eyes, and that's it, that little short range. Just to the right of that is the NIR spectrum. It is more than twice the size of light we can see with our eyes. If our eyes can tell us so much from such a narrow region of light, just imagine how much information is in the NIR region to be interpreted and understood. The mid-IR and far IR regions are even broader and can give us even more detailed information. However, the light in these regions do not bend, do not go far, and therefore require an instrument to be placed either directly on process equipment or very close to the process equipment. And the logistics of this are often very impractical. Now, just a little information on how the software running the instrument works. A clean sample, a clean spectra is analyzed and recorded. This is taken without the probe being in contact with any chemical, simply light going through the fiber optic cable, through the probe, and back through the other fiber optic cable. This is a baseline or background. Once this is done, the probe is placed in reaction material. The second picture here is an example of a sample spectra. The baseline is then subtracted from the sample spectra to show absorbances over the spectral range. The third picture here is an example of this, and it is this spectra which is then analyzed and yields, for example, component concentrations. NIR measures the changes in molecular bonds, specifically carbon to hydrogen, oxygen to hydrogen, and nitrogen to hydrogen. Most organic systems have these bonds. When hit with high energy light, these bonds bend, stretch, and do all kind of acrobatics that change the light. This is what the instrument detects and analyzes. Now that we've covered the basics, we finally get to the heart of this discussion, the advantages we have seen at my plant thanks to process analytical technology. Every time a sample is physically taken, there's a risk of chemical exposure. In this picture, you see a chemical technician dressed in an aluminized suit. This suit is worn for personal protection due to the pyrophoric nature of the chemical that he must sample for analytical purposes. This chemical not only ignites on contact with oxygen, but is so strong if it comes in contact with water, it will rip the hydrogen and oxygen apart and explode. Before process analytical technology, they used to have to sample this process 2 times a run. Now they only sample once at packaging. Another process we have uses a chemical which is immediately fatal to life and health if inhaled or absorbed through the skin. In this picture, you see 2 chemical technicians dressed in level A suits preparing to take a sample. The technicians in these suits are completely isolated from the environment they work in and breathe via supplied air. Once the sample arrives in the laboratory, 2 laboratory technicians would have to suit up similarly to analyze. When we first started this process, it was sampled once every 8 hours. After proving again and again that NIR analyses were accurate, we eventually were able to reduce sampling to twice a month. This was a serious sample reduction and a serious improvement to safety. Another advantage when reducing sampling of the previous slide was technician time and analytical consumables. To justify production charges that the lab had to make, this spreadsheet was used to show the analytical consumables and technician time associated with these tests. Each time this sample was analyzed, 2 technicians were necessary to stay with the sample until analyzed and cleaned up, a process which took about 2 hours. Dropping the samples to twice a month saved production considerable money on lab costs. The process with the pyrophoric chemical also saved money on lab costs once they began using PAT. They also saved so much time by eliminating that first sample that they were able to almost double their annual production, going from $36.6 million of profit to $66.6 million from one year to the next. The trend you see here is from a process that uses a very expensive catalyst to speed up the reaction. The reaction is component A, represented by the downward trending line, to component B, represented by the line at the bottom, to component C, represented by the upward trending line. If the reaction goes too fast, component B gets above 7% and heavies are formed which are hard to remove. The traditional analysis of this was done by GC, 1 sample an hour, and it took the lab about an hour to analyze and send the results back to production. If too much catalyst was added, it would be an hour before production knew, made changes, and another hour before they saw the results of their changes. To make matters worse, there were times when a needle would plug or there was some problem with the instrument and the analysis would be delayed. After monitoring with process analytical technology, the plant then received an analysis every 5 minutes, no delays. This resulted in better catalyst utilization, which means less money spent on buying catalyst, and better process yield. Production also saved on lab costs by eliminating all but the final sample in the sequence for verification and good manufacturing practices, as this was a pharmaceutical intermediate. Now, the two pictures here are the same process. The picture on the left shows all of the analyses done in a 24-hour period. 3 data points, which basically only tells the plant that they have met conditions and can proceed to the next batch. The picture on the right shows all the analytical of the same process after process analytical technology was installed. 1 sample every 90 seconds. 960 analyses in a 24-hour period. This data not only tells the plant they can proceed to the next batch, but it also let the engineers learn more about the reaction. By monitoring the trends, they can learn what happens when the moisture content of a raw material is too high. They have been able to get foreknowledge of the impending failure of a pump. They have also been able to reduce ride time, thereby increasing production. Now that I've shown you just a few of the advantages we have seen thanks to PAT, I want to give you a head start on justifying the purchase of your very own PAT system. I have learned from firsthand experience that no matter what advantages you may see by installing PAT, it's always a challenge to get a project approved by management whose main concern is money. These next 2 slides represent costs associated with a traditional GC compared to a NIR system. Now, these are figures from the last NIR that we purchased and a GC purchased the same year. They're approximately 5 years old and they're in US dollars. As you can see, the basic cost for a GC with all the necessary attachments is approximately $59,000. The total cost of a NIR system with all the necessary attachments is approximately $99,000. Most managers would stop you at this point and say, too expensive. However, this is just the purchase of the instrument. Also associated with GC are consumables. Helium gas, which gets rarer and more expensive every year. Nitrogen gas, compressed air, sample vials, solvents, syringes, and most expensive of all, technician time. Total cost first year for a GC, almost $92,000. Once the NIR is purchased, the only cost for the first year is the time necessary to develop the models the instrument will use to analyze whichever particular stream it's in. Total cost for the NIR for the first year, just over $100,000, but already the gap is narrowing. Your manager might still say, too expensive, even though the cost per sample for the NIR is significantly less than GC, because you get so much more information from the NIR. Now let's take a look at the second year. The second year, GC consumables and technician time stay the same, as they do for every consecutive year. The NIR, on the other hand, needs only a halogen lamp replaced. They usually last about 1.5 to 2 years. No technician time is needed because the instrument runs all by itself. The third year, GC costs remain the same. A laser may be necessary for the NIR, though newer technology is extending laser replacement times. The graph on the right should give you a good idea of the incredible savings of the NIR compared to GC over a period of 10 years. By the way, We just recently had to replace our first NIR that we ever purchased. It lasted for more than 22 years, running all day, every day. Anybody know what the average life expectancy of a GC is? Also, take into consideration that this example is just for one channel of the NIR. There are 5 more channels to use. However, since the basic system has already been purchased, any additional monitoring point only needs the purchase of fiber optic cables and a probe, a cost of approximately $18,500. For a second GC, you will have to pay full price. So what do you think your manager might say now? So here are some of the typical comments that I've heard management make with regards to purchasing process analytical technology. Top of the list, too expensive. I've already proven that statement is very narrow-minded. Close to the bottom of this list, you'll see no support. Now, I have to say that over the many years I've worked in the lab, there are a lot of companies who sell a little of everything, and this statement is sadly true for them. However, for those companies who specialize in process analytical technology, they have a great passion for the technology and their product. They are more than willing to help you better understand it, and they hope to infect you with their passion. If it were not for their existence, I would have never been able to go as far as PAT as I have. So reach out to these guys, talk with them, allow them to set up a demonstration. You'll be very glad you did. Now, with all the benefits I've shown you, I want to leave you with one thing to ponder and impress upon your memory. Ignoring the potential benefits of process analytical technology means throwing away real money. I thank you all for your attention. I thank Hellma for the opportunity to share with you my passion for PAT.
So thank you very much for your very interesting presentation. So I really like that you're not talking only about the technical side, but also about all the questions around. I think that's also very important to talk about.
Absolutely.
Really hope that you enjoyed and that you will share your experience and also take a look at our website to see when there is the next webinar. And please come back, join our webinars and talk with your colleagues about it. So thank you very much, Joey, for your presentation today. And thank you very much for the audience joining our session. Have a good day. Goodbye.
Thank you so much, guys. Y'all have a great day.